Professor Tony Cunningham, welcome to start day 2. Thank you.
So to counter some of the pessimism that we heard about yesterday, I thought I'd put some optimistic or over-optimistic views broadcast in LinkedIn. So current market size of the RNA vaccine market, several $billion dollars, still growing. Annual growth projected at 10% till 2033, an expanding pipeline of RNA and self-amplifying RNA vaccines for infectious diseases, cancer, autoimmune diseases, single-gene diseases. The major players, Moderna, Pfizer, BioNTech, CureVac with GSK, a market share, USA major, but Asia Pacific 15%. So we'll see how that plans out. We do know, however, looking at the past, that the development of vaccines has accelerated almost exponentially, and we all know just how quickly COVID-19 vaccines have been developed. So I'm going to review the fundamentals, look at the development history over decades, bust a few myths, and look at platform technology and the advantages and the challenges for RNA vaccines in the future. And just to remind everybody that researchers in the laboratory are well aware that naked RNA is quickly degraded and not efficiently delivered into cells. Shown in the left panel, it's rapidly degraded by RNases throughout the body. There is a lack of tissue tropism, and RNA by itself is not able to enter cells. So cometh the lipid nanoparticle, now well developed by most manufacturers, as well as other nanoparticles, which we haven't seen in licensed yet. But these lipid nanoparticles consist of 4 types of lipids. From the outside in, polyethylene glycol, which is hydrophilic, DSPC and cholesterol, which is structural, and this really important ionizable lipid, which helps the lipid nanoparticle get out of the endosome. The lipid nanoparticle by itself has weak adjuvanting properties, as well as also some assistance from the RNA itself. The lipid nanoparticles, which are produced by different manufacturers, differ in lipid constitution. One lipid nanoparticle is not the same as another between these manufacturers of Moderna, BioNTech, CureVac, and Arcturus, which produces the self-amplifying RNA. And myth-busting, the RNA vaccine, after it gets into the cytoplasm, is translated on the ribosome into cytoplasmic transmembrane or extracellular proteins that does not enter the nucleus and integrate into cellular DNA. And another myth buster, and that is that RNA vaccines have not been around just a couple of years before COVID. The developmental pathways to them have been going on for decades. So in lipid chemistry and pharmacology, the liposome RNA, even in the early 2000s, in immunology, knowing how to attenuate the inflammatory effects of RNA and the production of interferon over decades. In molecular biology, understanding how to engineer RNA, and microbiology, understanding how to code for the right type of surface protein, particularly the pre-fusion proteins, which I'll cover in a moment. The modifications to the RNA and vaccines have been substantial. Won Nobel Prizes for Drew Weissman and Katerina Katariko, and they consist of, starting at the left-hand end, modifications to the cap of the RNA responsible for initial binding to the ribosome, to the 5' untranslated region, to the signal peptide, and at the other end of the coding RNA to the 3' untranslated region, which increases RNA half-life, and to the poly-A tail, which increases translation efficiency and stability. And if we look at the coding sequence itself, numerous modifications have been introduced, mainly aimed at uridine, to try and reduce the capacity of RNA to induce interferons, which is not completely ablated, but to a trickle. We need a trickle for its immune stimulatory effects. But it also shows the modifications to the cap. And what about the synthesis of the RNA vaccine, now being done by a number of academic centres around Australia, not just by pharma and biotech. So once the sequence is known, synthesised off a DNA template, purified, and then mixed in large vats with the lipid nanoparticles, integrates very nicely, purified, and then fill and finish. And you can see this in some of the production facilities, both academic and commercial around Australia. A huge one now that Moderna has in this city. The COVID RNA vaccines differ in numerous ways. I mentioned the lipid formulation for the Pfizer and Moderna vaccines, but also the design of the non-coding structures, although these tend to be kept constant for an individual manufacturer, and also in the RNA dose. One of the roadblocks to RNA vaccines is that they're taken up by antigen-presenting cells into endosomes, and then the processing it continues with escape from the endosome, and this escape from the endosome is highly inefficient, only about 5 to 10% at best. So this is something that people, particularly at Monash, are working on. I mentioned before the modification of the surface or spike proteins, and this is probably well known to you with the SARS-CoV-2 spike protein, which has a pre-fusion and post-fusion conformation. And what does that mean? If you look on the left-hand side, the pre-fusion conformation is the conformation on the surface of the virion itself. And if you want to protect against infection, the antibody has got to be directed against that pre-fusion conformation, and it's like a trigger that when it enhances the receptor on the surface of cells, whatever virus it is, this stimulates fusion of virus and cell membranes, and in doing this, the protein changes conformation to post-fusion. And for many years, vaccines have been tried with post-fusion conformation. It's only really relatively recently we found ways to be able to stabilize the pre-fusion conformations. If we look at RSV, for instance, if you look at the top panel, which is the pre-fusion conformation, the bottom panel, which is post-fusion, you can see that the most important neutralizing epitopes recognized by antibodies on the proteins themselves are only seen in the pre-fusion conformation, the two left-hand ones in the top panel. And if we look at how the vaccines induce the immune response, you can see the sequence here of uptake of the vaccine into the endosome, escape, translation on the ribosome, and then cleavage of the protein on the proteasomes, presentation on MHC1 and 2, induction of B cells and CD4 and CD8 T cells. And if we look at how this occurs after intramuscular injection, the vaccine in the muscle encounters a fairly sparse distribution of antigen-presenting cells, macrophages and dendritic cells. However, the lipid nanoparticle and enclosed RNA rapidly transit through lymphatics to the lymph node, where they're taken up by peripheral macrophages and dendritic cells. There's then a cascade of interacting immune cells, which results in the production of antibody in the B cell follicles and activated T cells. And we're particularly interested in this, and you can see here is the slice of a whole lymph node. On the right-hand side, we're looking at a B cell follicle here, and patchy activation of macrophages after uptake, production of these cytokines, which kickstarts the whole cascade. This is with Shingrix, and Shingrix activates the inflammasome and the macrophages die. We have preliminary data with RNA vaccines now where they are also taken up by these cells, activate the inflammasome, but in fact they don't die. So we're looking forward to elucidating the differences in the immune cascades, which are so important in understanding the mechanism of action of these vaccines and trying to improve immunogenicity and reduce reactogenicity. Now, if we look at the biodistribution of RNA vaccines to bust a few other myths, vaccine antigen can be detected in the blood for only 24 hours after the first immunization. The spike antigen can be detected where you want it in the lymph node for 120 days. The mRNA from the vaccine persists in the blood for hours and elsewhere approximately for a month in some of the tissue organs. And biodistribution appears to be correlated with the type of lipid nanoparticle. Now, if we look at the advantages of the RNA vaccines, we've already talked about the rapidity and flexibility of production, and this actually applies also to experimental modification of vaccines. They encourage what's called reverse vaccinology, which allows scientists to rapidly fiddle with the sequences to get the right type of antigenicity. They're a platform technology. I'll come back to what that means in a moment. They can code for multiple antigens of the one virus. They can code for multiple viruses in the one vaccine, and there are new types of RNA vaccines such as self-amplifying. So platform technology. What I'm showing here at the ends of the RNA vaccine are the constant regions, the untranslated regions, which don't really differ for most of the vaccines of a given manufacturer, and it's really only the coding RNA that has changed. And so this has led to quite a lot of advocacy with regulatory agencies to look upon these vaccines as not completely new vaccines, say for RSV and flu, and will enhance and hasten the development and licensure of vaccines in the future. And just to remind you that most people would know just how quickly the COVID vaccine was developed. Firstly, the production of the RNA, then the production of the vaccine itself rapidly into trials within 65 days, and the emergency authorization or licensure within 11 months. And this allows RNA vaccines to be updated very rapidly, as we heard from Alan yesterday, moving from say, one SARS-CoV-2 variant to another, being able to produce the vaccines more rapidly so one can use the data from the latest strain surveillance from flu to produce the most updated vaccine and even perhaps target clade K. To remind you that the currently approved vaccines are the two COVID vaccines from Moderna and Pfizer, the RSV vaccine that we're going to hear about, and also in Japan, the self-amplifying RNA vaccine. And there's a very broad portfolio of vaccines being produced by several of the companies which I've already named. This is off the Moderna website that you can look at, the 3 licensed vaccines, other vaccines on the way for influenza and pandemic influenza, and at different stages of phase 3, phase 2, phase 1 trials, and also vaccines for herpes viruses, norovirus and HIV in development or trialling. Multiple companies are also developing combination vaccines that might provide substantial public health benefits. So flu and COVID, flu and RSV, all 3, or human metapneumovirus. And the only issue here, of course, is the timing of the epidemics, which we discussed yesterday, but obviously being able to produce, deliver 3 vaccines in one as we get more and more adult vaccines is going to be pretty important. Very important, of course, to make sure that there's no immunologic interference between the two or no enhancement of reactogenicity. And just to talk briefly about the last of these, and that is self-amplifying RNA vaccines. These use the replicative machinery of alpha viruses, flaviviruses, Venezuelan encephalitis virus to be specific. And in fact, this is not new again. Alex Kremik at the University of Queensland was experimenting with these very much in the early 2000s, as were others. The advantage of self-amplifying RNA vaccines is that you can put less RNA into the body, into cells, and the replicative machinery replicates the RNA prior to translation on the ribosome, and this may reduce the amount of RNA you need in the setting of a pandemic, for instance, and also reduce reactogenicity. And as I say, we've got one licensed, at least in one country at present. But there are future challenges for RNA vaccines: durability, reactogenicity, virus evolution, and we need to improve mucosal immunity to them to prevent viral infection and transmission. Preclinical models are not good. Immune-compromised patients, we still have problems. Safety, the rare side effects, we need to understand more about these, just as we do now understand the adenovirus induction of widespread thrombosis. And lastly, vaccine equity, which we did not see during COVID. So this just simply shows you the fall-off of antibody titers over 6 months, but T and B cell titers are retained in the blue lines with hybrid immunity. With infection, however, higher levels are obtained. And we understand now that, in fact, the best durable immunity is obtained from both infection and immunisation, which is a challenge, I think, in the future to how we might produce better vaccine boosters. We have similar data to this, and the T-cell data may be part of the reason why people are, in fact, getting longer-lasting immunity to severe disease, perhaps by T-cells in the lung. It's one of the weaknesses I mentioned is the inability of RNA vaccines to protect against infection and spread. So there are a number of academic centres and companies working to produce lipid nanoparticles that can be administered intranasally. And finally, the local manufacturing of RNA vaccines here in Australia by Moderna and BioNTech and in the future by Sanofi may not only help Australia but also the Asia Pacific, with particularly with the capacity to produce 100 million vaccines per annum as in the Moderna facility here. So in summary, the science behind RNA vaccines is decades old and continues to evolve. They use our body's protein production machinery. They're optimised at multiple levels to enhance delivery and production. The platform technology may help more rapidly develop and faster speed to market. The RNA platform allows de novo synthesis and updates, important for pandemics and rapidly evolving viruses like flu and SARS-CoV-2. This platform also allows vaccines against multiple antigens, for instance 6 in CMV, and against different viruses to be combined. And the nature of RNA manufacturing allows this to be set up quite easily in Australia and other countries. And of course, it's rapidly expanding. Thank you very much.
Sure. I've got one question online is, we're so good at making these, why can't we make a universal flu vaccine?
Well, that comes back to the design of the hemagglutinin and neuraminidase rather than the platform itself. And of course, the same is true of rapidly evolving viruses like HIV. And so
it's not the platform's fault, it's the virus's fault.
It's the virus's fault.
Yeah, that's right, blame the virus, that's right. John, got a question here?
Tony, great talk as usual. You tantalised us early by mentioning cancer vaccines, but you were singularly quiet on that. Can you give us a couple of comments? Are there? I mean, we've seen it with bladder cancer, we've seen it with some of the other cancers. What's the field? What's happening with that? And is there going to be anything clinical available in the next couple of months?
I think this is amazing. We're looking at individualised vaccines for cancer. This is an ID conference, so I wasn't allowed to talk about cancer. And it's possible to take 24 different RNAs, put them in the one vaccine, so you can sequence a person's cancer, look for the appropriate antigens, and now the immuno-oncologists are really good at detecting these and put them into the one vaccine. That's being done with melanoma, and in fact, if you use the current immunotherapeutic regimes and RNA vaccines, you can improve melanoma survival substantially. And that may be one of the ways in which we can get around some of the problems we've got with acceptance of RNA vaccines in the USA, because nobody is going to deny improved survival of people with cancer.
Okay, Alan.
So it's immune blockade, anti-PD-1, anti-CTLA-4, plus RNA vaccines.
So, Alan?
Oh, just wondering, I know you're talking about viral infections, but just wondering if you might want to, if you might be able to comment on the use of mRNA technologies for bacterial antigens and some of the considerations and limitations.
There are other people in the room much better at this than me. I mean, Jamie can talk about this with respect to TB, but certainly there are a number of RNA vaccines being developed for important bacteria, and TB is one of them. And again, this capacity to put multiple RNAs coding for multiple antigens into the one vaccine, I think, is substantial. Jamie might cover that in his talk.
Yeah, one more question here, and then the rest will take to the panel. Thank you.
Yeah, thank you, Catherine Satsky from the Murdoch Children's. I guess I was just interested in your comment around equity for these vaccines. It was, for those of us who work in global health, it was pretty depressing. And I guess just your thoughts on whether, you know, local manufacturing might provide some solutions for Australia to take a bit more of a leadership in this area, particularly in Asia Pacific.
So talking to Moderna about this, the original plan was that the plant here would supply not just Australia, but the Asia Pacific. 100 million doses a year is more than we need, and the work plans also for plants in Cape Town, for instance. I don't know what's happened to those plans, but it's, if you look at, I didn't show the BioNTech containers, but the encapsulation of this technology also may lead to much more flexibility in the future. And I think Africa has really got itself organised through its various organisations. So I think we'll see a better response in future pandemics.
Thank you.